In this section, we will discuss the hemostasis events in detail. So, let's start. First is the vascular constriction. So, immediately after a blood vessel has been cut or ruptured, the trauma to the vessel wall causes smooth muscle in the wall to contract. This instantaneously reduces the flow of blood from the ruptured vessel. The contraction results from the local myogenic spasm, local Autacoid factors, from the traumatized tissues, vascular endothelium, and blood platelets, and nervous reflexes. The nervous reflexes are initiated by pain nerve impulses or other sensory impulses that originate from the traumatized vessel or nearby tissues. However, even more vasoconstriction probably results from local myogenic contraction of the blood vessels, initiated by direct damage to the vascular wall. And for the smaller vessels, the platelets are responsible for much of the vasoconstriction by releasing a vasoconstrictor substance, thromboxane A2. The more severely a vessel is traumatized, the greater the degree of vascular spasm. The spasm can last for many minutes or even hours, during which time the processes of platelet plugging and blood coagulation can take place, moving towards formation of the platelet plug. If the cut in the blood vessel is very small, many very small vascular holes develop throughout the body each day. The cut is often sealed by a platelet plug, rather than by a blood clot. To understand this process, it is important that we first discuss the nature of platelets themselves. So, let's discuss the physical and chemical characteristics of platelets. Platelets, also called thrombocytes, are small discs, 1 to 4 micrometers in diameter. They are formed in the bone marrow from megakaryocytes, which are extremely large hematopoietic cells in the marrow. The megakaryocytes fragment into the small platelets in the bone marrow, or soon after entering the blood, especially as they squeeze through capillaries. The normal concentration of platelets in the blood is between 1 lakh and 50,000 and 4 lakh and 50,000 per microliter. Platelets have many functional characteristics of whole cells, even though they do not have nuclei and cannot reproduce. In their cytoplasm are the actin and myosin molecules, which are contractile proteins, similar to those found in muscle cells, and still another contractile protein, thrombostenin, that can cause the platelets to contract. In their cytoplasm are residuals of both the endoplasmic reticulum and Golgi apparatus that synthesize various enzymes and especially store large quantities of calcium ions. In their cytoplasm are also the mitochondria and enzyme systems that are capable of forming adenosine triphosphate, or ATP, and adenosine diphosphate, or ADP. In their cytoplasm are enzyme systems that synthesize prostaglandins, which are local hormones that cause many vascular and other local tissue reactions. In their cytoplasm are an important protein called fibrin stabilizing factor, which will be discussed later in relation to blood coagulation, and in their cytoplasm is a growth factor that causes vascular endothelial cells, vascular smooth muscle cells, and fibroblasts to multiply and grow, thus causing cellular growth that eventually helps repair damaged vascular walls. On the platelet cell membrane surface is a coat of glycoproteins that repulses adherence to normal endothelium, and yet causes adherence to injured areas of the vessel wall. especially to injured endothelial cells, and even more so to any exposed collagen from deep within the vessel wall. In addition, the platelet membrane contains large amounts of phospholipids that activate multiple stages in the blood clotting process, as discussed later. Thus, the platelet is an active structure. It has a half-life in the blood of only 8 to 12 days. So over several weeks, its functional processes run out. It is then eliminated from the circulation, mainly by the tissue macrophage system. More than half of the platelets are removed by macrophages in the spleen, where the blood passes through a latticework of tight trabeculae. Now, moving towards mechanism of platelet plug formation. This is endothelium, and this is basement membrane. Formation of a platelet plug in a severed blood vessel. Endothelial injury and exposure of the vascular extracellular matrix facilitates platelet adhesions and activation, which changes their shape and causes release of adenosine diphosphate, thromboxane, A2, and platelet activating factor. These platelet-secreted factors recruit additional platelets and aggregation to form a hemostatic plug. Von Willebrand factor serves as an adhesion bridge between subendothelial collagen and the glycoprotein IB, platelet receptor. But have you ever thought that what is the importance of platelet mechanism for closing vascular holes? Well, the platelet plugging mechanism is extremely important for closing minute ruptures in very small blood vessels that occur many thousands of times daily. Indeed, multiple small holes through the endothelial cells themselves are often closed by platelets actually fusing with the endothelial cells to form additional endothelial cell membranes. Literally, thousands of small hemorrhagic areas develop each day under the skin, such as petechiae, which appear as purple or red dots on the skin, and throughout the internal tissues of a person who has few blood platelets. This phenomenon does not occur in persons with normal numbers of platelets. Now, moving towards blood coagulation in the ruptured vessels. The third mechanism for hemostasis is formation of the blood clot. The clot begins to develop in 15 to 20 seconds. If the trauma to the vascular wall is severe, and in 1 to 2 minutes. If the trauma is minor, activator substances from the traumatized vascular wall, from platelets, and from blood proteins adhering to the traumatized vascular wall initiate the clotting process. The physical events of this process in case of severed injury of vessel are platelet aggregation, fibrin appearance, then fibrin clot forms, and then clot retraction. Within 3 to 6 minutes after rupture of a vessel, the entire opening or broken end of the vessel is filled with clot. If the vessel opening is not too large, after 20 to 60 minutes, the clot retracts, which closes the vessel still further. Platelets also play an important role in this clot retraction, as discussed later. Now, let's learn clotting factors in blood and their synonyms. Don't worry, I have prepared a mnemonic for you to learn them easily. Fibrinogen is factor 1. Prothrombin is factor 2. Tissue thromboplastin is factor 3. Calcium ion is factor 4. Labile factor is factor 5. Stable factor is factor 7. Anti-hemophilic factor is factor 8. Christmas factor is factor 9. Stewart-Prower factor is factor 10. Plasma thromboplastin antecedent is factor 11. Hageman factor is factor 12. Fibrin stabilizing factor is factor 13. So, an easy mnemonic to remember these clotting factors is the fit pants, tight collars, loose shirts, are cool, says pretty, heroine, Farah. Let's repeat this mnemonic with me. Fit pants, tight collars, loose shirts, are cool, says pretty, heroine, Farah. Now, moving towards the last event, and that is the fibrous organization, or dissolution of blood clot. Once a blood clot has formed, it can follow one of two courses. It can become invaded by fibroblasts, which subsequently form connective tissue all through the clot, or it can dissolve. The usual course for a clot that forms in a small hole of a vessel wall is invasion by fibroblasts beginning within a few hours after the clot is formed, which is promoted at least partially by growth factor secreted by platelets. This process continues to complete organization of the clot into fibrous tissue within about 1 to 2 weeks. Conversely, when excess blood has leaked into the tissues and tissue clots have formed where they are not needed, special substances in the clot usually become activated. These substances function as enzymes to dissolve the clot. That will be discussed later in this video. This was all about this section. In our next section, we will talk about the mechanism of blood coagulation.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.